session is Joao Seco, professor and division head of biomedical physics in radiation oncology in DKFZ, the German Cancer Research Center. Joao, please go ahead.
All right, good afternoon, and I would like to thank the organizers for inviting me. It's a pretty broad presentation to give an overview, a bit of the cancer biology, and then I get into at the end on two topics on flash and mini beams. At any point, if there are any questions, you're welcome to interrupt, to ask the questions. It is a bit long, so that's why if you wish to ask any questions, please do interrupt. Okay, so let me see if I can get this to move. Okay, so when we treat cancer, we have to remember that the most important thing is to try to find a way of finding the weakness in the cancer. So this is the so-called holy grail of oncology. We want to try to find a so-called monotherapy, something that separates the cancer from the healthy tissue around it. Let me just bring up the pointer when I need it. Okay, so that is usually very hard to do. But that's usually what we try to do. So, and when we treat, of course, we want to reduce as much the side effects if possible. Okay, so a little bit of biology. So we have to remember that the cells, they just have, some of them have a special property, which is the so-called stem cell. And the stem cell is the one that has the ability to renew itself, to keep dividing and to produce more cells, right? And we need stem cells in order to exist. So we are constantly renewing our cells. And then the second step is that when the stem cells, they transition, they become more specialized. And so they become these so-called specialized cells, and eventually they die off after 3 or 4 generations. So here is 4 generations, and eventually the cells die off. And so they tend to then get replaced by new stem cells that replace the so-called specialized or differentiated cells. And let me just switch off my. Okay, so back to the presentation. Something that is really important that people forget is the flow of information in the biological system. So the DNA is the kind of central library. It accumulates all the information that the cell needs to exist and to survive. And the way the DNA interacts with all its components is through something called RNA. So it makes copies of the DNA, so-called RNA copies. And it is then from these RNA copies that it makes the so-called proteins. And it's the proteins that allows the cell to interact with whatever stress or situation that it needs. So this is a 2-step process, where the first one is called a transcription. The DNA is copied into an RNA. And the second step is the so-called translation. The transcription happens in the nucleus where the DNA exists, but the production of proteins happens in the cytosol. So this RNA has to exit the nucleus and go into the cytosol to get produced to allow the cell to respond to any type of stress. So this is the way the information is processed in the cellular level. So how do we diagnose cancer, right? So that's one thing that is really important. Well, there's a variety of ways of doing it. We can either measure very small molecules through either genomics, proteomics, or metabolomics, or cellonomics. This is at the molecular level. We can also study the cells, or in a more macroscopic way, the way we do it in medical physics, is we could use either PET or MRI or CT. Here, for example, on the right is an example of a PET CT patient, where you see the PET activity in the bladder and in the brain for the sugar, and you see the CT in the background. So there's different ways of diagnosing. But when we do with patients, we tend to use these macroscopic images. And then we go back and do analysis of these smaller properties like the cells on the molecule. So when a clinician looks at a cancer, he also wants to do what is called a biopsy. He wants to look at the cancer directly to understand what is the pathology, right? So there are 2 types of biopsies. This is the so-called liquid biopsy, where you can take a fluid or blood sample and you analyze the blood sample. Or you take a solid biopsy and you go in with a needle and you remove a small part of the tumor and you analyze it. And then the analysis involves a variety of steps. You can either look at it under a microscope with an H&M dye, for example. That's your normal pathologist will do that. You can also do a proteomic or genomic profile where you look at the protein activity and the gene activity of the tumor. So these are different ways that the clinician can analyze what type of cancer is present to have a feel for it. So keep in mind that cancer patients are usually very sick, and so the physical biopsy is really not so common, but the liquid biopsy is relatively easy to do. Okay, so what does the pathologist look at when he looks under a microscope? Well, here is the difference between a healthy cell lines, healthy cell environment, and on the right, you see the case of a cancer. So the first thing that the pathologist looks at is the structure. Is the cells in a very organized, same shape, same structure? And if that is, it's a good sign. If it isn't, it's cancer. And you see that the shape, the size, even the differentiation is completely different. I have 4 different types of cells here, while here I have 4 identical cells. And the morphology and the shape changes from the cancer to the normal. And the other thing that is pretty common is that the tumor is usually completely disorganized compared to a healthy tissue. So I will go into this in a second. Here is an example of how cancer looks like when you grow it in the microscope. So it has a healthy set of cells, and you see the structure is very organized, very well defined. And as you start to have hyperplasia, you start to see small pressure and growth. And again, you see more mild dysplasia, and you start to have a complete chaos. And then eventually you can have the so-called extremely aggressive cancers that start to metastasize. They start to move around, and eventually they can even infiltrate blood vessels. So they drill a hole in the blood vessel and they jump into the blood. All right. So these are the invasive cancer, this one here on the right. This is a cancer that is progressing towards becoming invasive. And here you have your low-grade cancer, which is the mild dysplasia, but you have disorganization because of it. So the key idea is that you have to remember that the cell has grown the ability to get unlimited division. So it is kind of growing without stopping. And this is only possible because some of the cancer cells have the so-called stem-like properties. They can have infinite division without ever dying. And so the cancer can spread because of this rampant growth. And it allows it to spread, of course, right? The metastatic spread is something completely different because in addition to growing in an unlimited fashion, it also grows the ability to drill holes in blood vessels. Okay, so this is not so easy to do, but it's possible. So here's the example of a normal division of a cell. Here you have a division of a cell, you produce a few more cells, and when they die, they go through something called programmed cell death. And the programmed cell death mechanism is a way that a cell dies in a way that it doesn't affect the body. In the case of cancer cells, they find ways to evade the so-called programmed cell death. And one way is to inhibit a gene that switches on this programmed cell death. There's a variety of genes that do that. And the most famous one is the so-called p53 gene. And so they can basically dodge a bullet, effectively. They can just keep going on and on and on without any trouble. And you also notice the differentiation. All these cells are completely different, yeah, compared to this one here. So as you have these mutations, the cells start to adapt and to change, and they allow them to mutate and move around and even become extremely aggressive. So the mutations are something that happens with the cell division, and the mutations allow them to jump or skip cell death, and at the same time allow them to proliferate. All right, so now one thing that is really important is that when a clinician analyzes a cancer patient, he or she wants to understand if the patient has a low-grade cancer or a high-grade cancer. The cancers are usually scored in 4 grades, 1, 2, 3, and 4 in Roman numerals. Low grades are usually 1 and 2, and high grades are 3 and 4. The big separation between the 2 grades is how they metastasize. So high-grade cancer is the one that has metastasized, and the low-grade cancer is the one that has not metastasized. So the big difference is low grades have not been able to drill holes in blood vessels to allow them to spread, and the high grade have already been able to do that. So you see a huge difference in survival. So here on the left, on the y-axis represents the patient's survival, and this after 5 years. This is your standard procedure for analyzing patients after treatment. And you see after 5 years, the low grades have a very high survival rate, usually in the 80s to 90%. while the low grades have a very high survival, while the high grades have a very poor survival at 5 years, usually around 20%, sometimes 30% survival. This is a factor of usually 2 or 3. So it's a very poor survival rate. And this is what radiotherapy tries to do. It tries to improve the survival rate. Okay, so let's come a bit to cancer statistics. So the statistics I'm presenting is from 2017, so it's 4 years ago. And the first graph represents the so-called cancer incidence, all the new cancers that appear every year. And you see the dominant cancer incidence for men is prostate and lung, while for female, it's breast and lung. So you see a huge difference, right? For men, it's prostate, for women, it's breast, and lung is common for both. And the two top cases are almost 40% of the patient, 30% on the man and close to 40% on the woman. So it's a very high level cancer incidence. So cancer incidence means new cancer. The cancer deaths is the amount of people that die from the cancer. And at the moment, the highest cancer killer is lung in both cases. So lung is almost 30% for males and 25% for females. It's a very huge impact. So that's why lung has become so-called public enemy number 1 for cancer. It's really taking up a lot of life. And most of this lung cancer is related to tobacco. So you really have to pay attention to that. Okay, here is the spread of the cancer incidence. And as already mentioned in previous talks, the cancer incidence increases with age. So the longer you live, the more likely you are to have cancer. So cancer is an old age type situation, old age disease. So you see here in North America and in Europe and Australasia that the largest cancer incidence are the countries with high or long-lived life. So Japan, of course, some Southeast Asia, Australia have an extended life, and they have the ones with the highest incidence. And then you have a lower incidence in Africa, of course, because they're not living as long, and then you have less in Russia for different reasons. Okay, so I've spoken a little bit how the clinician looks at a cancer, but I've really not told you what's really one of the main driving forces of cancer. How do we know something is cancer, right? So there's something called the Warburg effect that is at the heart of cancer, and nearly every cancer has the so-called Warburg effect expressed. So it's all got to do with the way we produce energy in the cell. So here is a cell in orange. This is your normal cell. And this little red dot is sugar, right? So the sugar has been delivered to the cell from the outside. It enters the cell and eventually it will produce so-called energy. So the mitochondria is the so-called power plant in the cell, and it produces energy called ATP. So ATP is the energy that we need for anything to function in the cell. And it's a two-step process. The first step, it breaks up the sugar into 2 molecules, so-called pyruvates. So the sugar is a carbon-6 molecule, all right? And the pyruvate is a carbon-3 molecule. So you see you have 1 sugar leads to 2 pyruvates. And eventually then this pyruvate will enter the mitochondria and with the help of oxygen produces energy. So this whole process from sugar to ATP produces roughly 40 ATP, 40. Okay? So it's a very, very efficient way of producing energy, and it requires the so-called two-step process, where most of the energy production is done in the mitochondria. So of the 40 molecules of ATP, I produce 38 in the mitochondria. The other 2 are produced in this step here of the pyruvate. So this part is called oxidative phosphorylation, and it is the conversion of sugar into energy, which is the ATP. Many years ago, a famous chemist called Louis Pasteur discovered that if you switch off oxygen, right, you have something called glycolysis. So you switch off the second component and you produce energy just with this second step, the pyruvate. And this is very well known because it's what we use to produce alcohol, or it's the way we use it, it's fermentation. So here, the Pasteur effect happens in the presence or the lack of oxygen. So there is no oxygen present. And you produce all the energy here in the pyruvate, and at the same time, you're fermenting any type of thing that you produce. And you produce a lot of byproducts, which is the so-called lactate, which is an acid, which is immediately ejected out of the cell. So you see the two extremes. You have oxygen and you produce a lot of energy. You have no oxygen and you produce energy, but you produce a lot of this byproduct, lactate and so on. So the big difference in energy is 40 on the left, 2 on the right. So it's highly inefficient. So what happened? In 1910, 1920, a famous chemist called Otto Warburg decided to investigate why cancer cells became cancerous, and he discovered the so-called Warburg effect. And the funny thing about this whole thing is that I can produce energy through this glycolysis effect in the presence of oxygen. So this is very different of the Pasteur, where there was no oxygen. The cancer cells, in the presence of oxygen, still do not use the mitochondria to produce energy. They prefer to use only the glycolysis step, which is the first step. So this is usually called the Warburg effect, the so-called aerobic glycolysis, energy production without the mitochondria, even in the presence of oxygen. So this is the question that's been around for over 100 years, and no one has been able to answer this question. Why do cancer cells produce energy in the presence of oxygen without using the mitochondria? And this is a big problem because it also makes the cells very, very resistant. So the oxygen element that was supposed to be here is a very important sensitizer for radiation. And if the cell doesn't need oxygen, it makes it extremely resistant to therapy. So these cells, which have the Warburg effect switched on, are highly resistant to radiation. Okay, so let's look at the different types of ways to treat cancer. Here are more or less 3 pictures representing 3 ways. One of them, which is the most common, is surgery. A clinician will go in and surgically remove the cancer, so you have the removal. A second one is administration of drugs in the form of chemotherapy. So you inject or you swallow some drugs and they go and deal with the cancer all over. And a third case is the radiotherapy, which is the one we're going to be talking about. And so you use a variety of radiation beams to treat a specific localized cancer. So there are 2 types of approaches when we use radiation. One of them is to cure the patient. So this is only possible in low-grade cancers, grade 1 and grade 2. We go in and we want to cure the patient. And the palliative case is when the person has no chance of survival to the cancer. And what we want to do is improve the person's quality of life. So this is primarily driven to grade 3 and grade 4 of the cancer, where we are just going in and improving quality of life, and in many cases, even reducing pain. So chances of survival depend a lot, but on average now, in the 2015 report, we have around 65% survival for all cancers overall at 5 years. So it's improved significantly. It used to be around 50%. So it's it used to be around 50% in 1975. 30 years later, it's improved by 15%. So it is much, much better. Still, there are extremely resistant cancers. Pancreatic cancer, for example, has extremely poor prognosis. It is considered the hardest cancer to treat, while others like prostate and breast have done extremely well. So there are different types of radiation therapy methods. There's the so-called external, there's the internal, and the external has a variety of methods: photon therapy, particle therapy, and of course, the internal, you have also the brachytherapy. So one thing you have to remember is the amount of patients that go through radiotherapy. So in the last estimate in 2020, there were roughly 10 million patients, new cancer patients every year, 10 million, of which 50% will receive radiotherapy. 50%, that's at least 5 million receiving radiotherapy. And so the radiotherapy, the way it works, is that it eventually produces damage in this DNA. So here is your cell picture. You have your nucleus here where you have your DNA, and the radiation will eventually damage the DNA enough for it to stop growing and eventually to die. So I'm not going to go into the radiochemistry. I'm just going to jump this part of the radiochemistry. Here's a little bit about particle therapy. This has already been presented in a previous one. So I just want to mention some important dates. The idea started in 1946 by Robert Wilson. In 1954, approximately 8 years later, they treated the first patient in Berkeley. And the first hospital facility was in Loma Linda, which is here, 92. And then there was a variety of other centers open all over the world, including Europe, Heidelberg, Knaup, and Medalstrom. Okay, so I want to bring up something that Robert Wilson mentioned in his paper, which is really important to remember sometimes. is that in this visionary paper, he proposed many things. He proposed that protons can be used clinically, so that's a good. He proposed that accelerators are available. Yes, that was also possible through Lawrence Berkeley. He also proposed that you could place the maximum radiation dose in the tumor. Yes, that's using the Bragg peak. But he proposed also that protons should spare organs. This is kind of a big jump, right? So you want to use the radiation to spare healthy organs. It wasn't so easy at the time. And another thing that he proposed is to use modulator wheels to spread the Bragg peak, to create a so-called spread out Bragg. So let's look at how it used to be, right? Radiotherapy in the particle center used to be like this. It used to be almost like a physics laboratory. So you see the patient here, and you see this almost like a lab in the physics department where you're putting things together. This used to be what used to be around in the 90s or even before. This is now modern radiotherapy, particle therapy in a proton center. So it's completely different. You have to remember that the patient is usually very sick. You want to be able to give the person a very quiet environment that she can get, can be treated, and you want minimal fuss around the whole problem. Okay, so let's quantify the true advantage of protons over photons. Here is your so-called Bragg peak in the blue dotted line, and the yellow represents your X-rays. And you can produce the so-called blue line here by adding a few of these Bragg peaks. And this is your so-called spread out Bragg peak. So one advantage is immediate. You have a low entrance dose, and this allows you to really reduce the radiation at the entrance. You have your maximum peak that was proposed by Robert Wilson. You have your rapid fall off of the dose right here. And you have your energy modulation that produces the spread out Bragg peak. And you have approximately an RBE of 1.1, which is very similar to photons, which is excellent. Okay, so now how do we really say the true advantage of protons are this and relative to X-rays? So you have to remember that the key idea is that if I irradiate any tumor cell or any cell with radiation, the more radiation I give, the more likely I am to kill 100% of the tumor. So if I increase the radiation to the cell, eventually I will achieve what is called 100% tumor control. It doesn't matter what cell it is. So if I give 100 or 200 or 500 gray, eventually I will destroy the tumor. The big problem is the healthy tissue. I cannot keep increasing the radiation to the tumor without having side effects on the healthy organ. And here's an example of a photon curve. And the small shift between the two of them is effectively related to DNA repair of the cancer. Usually the cancer has a worse DNA repair, and so you have a small advantage between the two of them. So when we treat with photons, we would always like to be in the so-called benefit region, here where you have maximal effect. So here is more or less the point at which you have maximum tumor control with the minimal so-called toxicity. In reality, the clinicians are extremely conservative, and you're really very low. And you are at the level of 20–30% toxicity to achieve more or less 30% tumor control. So you see in this case, most of the cancer is survived the radiation. So it's really not a very successful treatment. So now enter protons, or particles. The big advantage of protons is that I can reduce the dose to healthy tissue. So effectively, I move this curve to the right. And for the same level of toxicity, I achieve a much, much higher level of control of the cancer. So now I'm in the 90% control. So notice I went from 30% to 90%. This is truly the advantage of protons and particles. The fact that I can reduce the toxicity of my treatment means I can really dose escalate to the tumor and achieve better control. This is the most important figure of my presentation. If you understand this figure, you understand how particles work. All right, so you've already seen this figure. In the previous talk, you see there was an exponential growth of particle centers in the last 10 years, and you see them growing, and now they are over 200. So it's really amazing. I will not get into this. I will jump this part. I will talk a little bit about generation of photons and particle beams, because this is important for the rest of the talk. So one thing is to remember that photons are usually generated through a thermionic emission. So I have electrons that are emitted from a cathode. They are then accelerated to an anode, and I produce these electrons, which I inject into a linac. right, in the linear accelerated tube, and I eventually produce X-rays. And here's an example of an X-ray machine that is being used for treatment. I have my linear accelerated region, I have my electron gun, I have my target up here, and I have all my collimation system up here, and I produce X-rays. This is very compact. Notice how compact this is, very small in a small room. This is the production of X-rays. In the case of protons or carbon ions, you need an acceleration structure. Either you use a synchrotron, which has already been discussed in a previous talk, or you can use a cyclotron. Here is the HIT facility. It has a synchrotron and it produces 4 types of ions. It has the protons and the helium, and it has the carbon and the oxygen ions. And that's a facility that allows you to accelerate these 4 ions for therapy. So you have your racetrack here, your synchrotron, you have 2 fixed beam lines that go in this way, and you have a very large gantry for therapy. Yeah, and you even have a 4th line for experimental research. I'll jump that. So now we come into the part that is important, the delivery. There are 2 main methods of delivering protons. There is so-called passive scattering versus pencil beam scattering. The passive scattering system is the way to build a SOBP. I have my SOBP that's made up of multiple Bragg peaks. You see the intensity of each Bragg peak produces a small region where I'm going to treat the cancer. And to produce this with a passive scattering, I need something like a range modulator wheel. So here's a range modulator wheel where you can see the steps. And each of these steps is approximately 3 millimeters in range, different from the next one. So there are 3 millimeter steps. Or you can have a ridge filter. And this allows you to produce multiple Bragg peaks with a slight shift in their range. And you can produce many of them. And in order to do that with a very long beamline, you have your range modulator wheel up here. You have then your double scattering system, which amplifies the fluence profile. And then you have your range compensator here. You have an aperture and a compensator here. And you produce any shape of treatment. So the passive scattering beams allow you to shape the distal part of your tumor. So notice that the distal part of the tumor, which is this part here, is perfectly shaped with the compensator. You lose the ability to shape the proximal part where you have these horns. You usually have these horns in passive scattering. So, so the passive scattering system was the first one that was developed, and it's been around for over 30 years. Pencil beam scattering is an alternative. So here is my passive scattering. I shape the distal part. I have the horns. In pencil beam scanning, I have basically like a television. I can basically spot any region and produce any shape of dose distribution I want. And so the horns have disappeared. I've completely lost them, and I shape any type of organ tumor that I need. So the big difference is the hot areas that I had in the passive scattering is now gone, and I have a perfectly shaped tumor. Okay, so how do I deliver protons? I used 2 types of fields. I can either use a so-called single field uniform dose, or SFUD, and there I deliver a spread out Bragg peak, which is uniform, as I demonstrate here on this top right figure. And like this, I create 3 beams, for example, and I generate a uniform dose distribution. An alternative way to deliver protons is to use intensity modulated proton therapy, where now I don't have a uniform dose distribution, but I have a modulated fluence distribution. So now I modulate this fluence here to achieve any shape I need that allows me to protect organs that are close by. In both cases, I will deliver dose distribution to the tumor while reducing as much as possible the radiation to the organs at risk. Okay. The big difference is one is uniform, one is non-uniform. Here's an example if I compare IMPT with all the other techniques out there. So the top line are the photon techniques and the bottom line are the proton. And you see the top line, the 3D conformal, I have a very big hot area, and I also have all the other organs receiving radiation. The top right corner is IMRT, and I have much better shaping of the tumor, but I still have a low dose all over the whole body. Then with protons, the left bottom curve is the passive scattering, and I see I have a better shape of the tumor, and I have less dose to the remaining organs. And finally, if I use IMPT, I can even reduce significantly the amount of radiation I give to all these different organs. You can see that clearly here in the spinal cord, you have a lot less dose. For sure in the lung, the dose is reduced, and you have the ability to produce any tumor shape that you want. So these are the main differences. I just want to highlight the two methods together. Here is the IMRT on the left with photons and the IMPT with protons on the right. And this is an extremely sensitive region of the brain stem. And you see that with the protons, I can literally reduce the dose here to practically zero. While here, with IMRT, I'm still giving a good amount of dose, 20% of the total dose. So this is a big advantage of IMPT. I can reduce the dose to organs very, very close to the tumor to very, very low, reducing side effects from the therapy. Okay, here are the different types of Bragg peaks for different types of ions compared to the photon. I've already shown you the photon to protons. I haven't shown you the carbon ions yet. So the green line is the carbon ions. They also have a Bragg peak. The Bragg peak is much sharper, and it produces a very large fragmentation tail, depending on the energy of the carbon ions, of course. So here's the key slight, let's say, disadvantage. This fragmentation tail does create a lot of biological dose, and you have to pay attention with it. Here is the same from a 2D dose distribution. You see the photon curve produces dose all the way through. The protons has this dose here that ends at the distal part. And you have this fragmentation tail here producing additional dose. And this is just physical dose, but when you do carbon ions, you really have to account for this biological effect in this end of range, in this fragmentation tail. Okay, so now what's the advantage? Why bother with carbon or oxygen ions? The key idea is the damage. While in protons and photons, you tend to only destroy one part of the DNA. So remember that the DNA is a double helix. That means there are always two sets of copies of the DNA information. So you see this by the two little balls, yeah? There are two copies of the information for each DNA. You take out one of them and it can repair based on the other copy. So I lose the red one here and I lose the purple one, it can repair. And here I lose another red one and so on. And this is the case that works for low LET particles like photons and protons. I tend to only remove only one part of the information, and it allows the cell to repair and to recover. In the case of carbon, I destroy both sets of information. There is no way of coming back from that information. I don't have a copy from which I can rebuild the DNA. So in that case, carbon is lethal, while protons and photons are so-called potentially lethal. You have to really give a very high dose to guarantee a lot of the DNA is damaged beyond repair. Here's an example of it. On the left side, you have a photon beam. You see the uniform distribution of radiation dose deposit. And here on the middle and the right, you start to see the track structure as you start to increase the LET. So you have, for example, a proton on the top. You can have a carbon ion in the bottom one here. This is just so-called carbon ion. And you can have a very, very heavy ion like gold or anything else. And you see the huge amount of track structure dose plus subtract. So you have a subtract here that produces an additional damage. So this produces complete damage that destroys all the DNA information that is present in the section. Okay, so now I want to enter one of the hot topics, which is called flash. I think it's been mentioned already this morning, and I will provide some extra details and maybe even answer some of the questions that were asked about flash this morning. So flash radiotherapy was an idea that's been around already since the 60s. And I have a picture of the people that worked on it. So in 1967, the first flash study was published for HeLa cells, which is mammalian cells. And then there was a spree of papers, 69, 70, up to 74, all of them for different types of cells. So mammalian cells, animal cells, these CHL or Chinese hamster lung cells, and a variety of E. coli bacteria also. These are the studies that were done in vitro. And recently there was a few in vitro studies done in 2019 by Adrian and Bonanno from Memorial on lung. But we also have a lot of flash studies in animals. And so there was not so many animal studies done in the 60s and 70s. There are two here, but there was a significant amount of animal studies done in the last 10 years. The most famous one that started everything is the Fovedon paper in 2014. And they published the first paper that basically relaunched the whole field of flash. And there are a variety of models out there. And I will discuss these models in a minute, but a lot of the studies varied between experimental model. So the papers were published with an experimental model and a model. While most recent studies, you had usually a separation between the experimental study and the model study. So which was a bit odd, but different. Okay, so what is the main model that is used at the moment to explain FLASH? It's called oxygen depletion. Oxygen depletes and the FLASH then ends up being protective. And this was initially proposed in 1967 by the Town paper, and a few other papers came along to kind of substantiate this idea. While in all the studies in the last 10 years, they've all based on oxygen depletion. So you see there's about 10 papers here, all of them based on that. Then there were a variety of other models, something called radical recombination and liquid peroxidation. All these models were alternative models that were proposed to try to explain FLASH. I want to bring to the attention that no one really knows how FLASH works. These are unproven models. And so there was a variety of many papers, in fact, based on really unproven models. So here is the key idea. The oxygen enhancement ratio is this one I represented here on the left, and it represents how oxygen sensitizes cells to radiation. So it's usually defined as something that measures the effect of oxygen from 0 oxygen up to 21% oxygen, which is your up here at the 140 millimeters of mercury. So this is your oxygen pressure, and this is the damage. So at 0 oxygen, I usually reference this as to 1. And as I increase the oxygen, I sensitize the cells to radiation, and I increase the factor 3. So it goes from 1 to 3 cell kill. So in the presence of oxygen, I kill 3 times more cells than without oxygen. This is a major difference because this is the key factor that allows clinicians to understand, is the cancer resistant to radiation or not? So that's usually what they do. When the clinician wants to study a specific cancer and he wants to treat the patient, the first thing he wants to do is to understand if the cancer is resistant to radiation. And that usually means, is the cancer hypoxic? Hypoxic cancer are very resistant because it's not sensitive to oxygen damage. The first FLASH study, well, the second one, 1969, they did is compare radiation effects on cells. And they compared both animal cells, hamster and HeLa cells, and they found that in the presence of normal radiation, you would get a very high cell kill. So this is a so-called clonogenic survival curve, usually given in log scale versus dose. So this is a log scale. And in this case, you have a normal, the white dots represent your normal clonogenic survival curve. And the FLASH effect is the one that's the black line. So you see here that for normal dose delivery, I kill a lot of cells, but under FLASH, I spare the cells a lot. So there's a huge decrease in the number of cell kills. So for the same dose, 10 gray, I kill 99% of the cells. In this case, here I only kill 10% of 90% of the cells. So it's a big difference. And the same thing was observed for HeLa cells. And the previous study by Town in 1967 saw that the behavior was as if the cells were behaving hypoxic. So here's your normal cell kill, and this is your hypoxic survival curve, the one in nitrogen. And if you irradiate under FLASH conditions, you suddenly have the so-called inflection point here in the middle. And from there, you have a hypoxic behavior of cells. So this straight line represents the cells that are under FLASH. So that's why the oxygen depletion has become so important. People started to understand that the cells in the presence of FLASH radiation behave as if it's hypoxic. And that means the cell goes from behaving at a factor 3 to 1. So you have a factor 3 decrease in cell kill. So this is a major decrease and allows you to spare organs, of course, if you're irradiating them under FLASH. So here's the study from 1967. I want to compare that to a study that was done very recently in 2019. And you see also the difference in the quality of the data. I clearly see a very clear inflection point here that is not available in this data here on the left. So the blue represents the FLASH, the red represents the standard curve. You see a small difference here in the oxygen, but you don't see a clear inflection point here between the two of them. So that's a major difference. It's amazing that the biological data in the 70s was extremely good. They spent a lot of time, while in the last recent years, the data has been really difficult to analyze. So here is an overview of all the results that came out in the 60s related to FLASH. And there was a huge difference in the way they treated FLASH. They first gave a minimum dose in the FLASH treatment, so around 5 to 12 gray in one single pulse. And the pulse rate was, the dose rate was 10 to 7 gray per second. So it's a very, very high rate. So I'm giving almost 10 gray in a microsecond effectively. So you have to remember that it's amazing the amount of dose I give in a single shot. And all these found that the so-called inflection point required a minimum dose between 5 to 12. Okay, so now let's come to the famous paper in 2014, where they saw the FLASH protection in normal tissue. And the Forvidon paper, what they did is they compared a 4 MeV electron, 4.5 MeV electron with an X-ray beam irradiating mice. And they immediately found that there was a huge response in the way they irradiated lung. So the red curve represents your standard lung treatment with 1 gray per minute. The blue curve represents the same mouse, but receiving at 3,600 gray per minute. So that's basically 40 gray per second. And that's a huge difference. And on the vertical axis is pulmonary fibrosis. That means that the lung has a huge side effect, which is the fibrosis effect produced by radiation. And you see that there is a significant difference in pulmonary fibrosis between the mice irradiated with FLASH compared to the mice that were not irradiated with FLASH. The radiation was producing a huge difference in side effect. FLASH was doing this. And when they looked at the histology, they found that a 17 gray conventional had a huge amount of side effects. You can see this is called an HNE plot. And you see, you see that the damage is completely clear. But if you compare 17 gray FLASH with a normal control, you see there is not a big difference in the histology. So they look very good and they look very similar. While this one is, the cells are in very, very bad shape. So the darker the color, the more signaling it tells you that the cell is dying. So the H&E plot basically tells you that if it becomes darker and darker, the cell is dying. So you see there's a huge amount of dark purple, which the cell is entering into cell death mechanism. So recently, in the last 5 years, there was a review paper that looked at all the accumulated evidence of FLASH. And these are from all the recent studies, and the review paper was published by Vosna and al. And they looked at different dose and dose rates. And the dose rates were varied between 15, 17 gray up to 100, 300 gray per second, sorry. While the minimum dose was always extremely high, a minimum of 10 gray was the shot dose. So you see that all of these showed evidence of FLASH protection. So mouse intestine, mouse skin, mouse tail, mini pig and cat, mouse lung, mouse brain, all of these show FLASH protection. So there was a good amount of indication that it's clinically there. And these are all animal studies, not patients. So one of the first FLASH systems to deliver FLASH was the IBA system in Groningen. And what they showed is that a proton facility with a cyclotron can deliver FLASH. This FLASH treatment was not done with a patient. It was just done with a dosimeter. And they showed that they can achieve 100 gray per second in using a normal cyclotron for proton beam. So this was an amazing thing, and it was done 2019. So it was March 2019, more than 2 years ago. Okay, so we also spent the last year and a half working on FLASH to understand, is oxygen the key driver? Is oxygen depletion the key driver of FLASH? And we just recently got a paper published that I wanted to mention. And what we did is we decided to measure directly the oxygen that is present in the medium and how it's affected by FLASH. And so we used what is called an infrared sensor that we put on the inside, right next to the medium and next to the cells. And we used that infrared sensor to tell us the amount of oxygen that was present. And it's a tiny little probe that you can see here with this little red dot, black dot, that represents the probe that we are basically measuring oxygen. So it was a simple way to check, is oxygen needed or is oxygen present? More important was, is oxygen being depleted during radiation? So we did these measurements for a variety of radiation types. We did that for carbon and protons on one side, and we did it for X-rays on the other side. And we tried a variety of dose rates. And in the case of X-rays, we managed to reach up to 50 gray per second. So that's definitely in the flash range, where it's usually considered to be flash with 40 gray per second. And you see the dose rate represents different levels of oxygen being removed. So on the vertical axis, you have your oxygen concentration that is present. And on the x-axis, you have your time. And so the faster rates remove oxygen very quickly. So you see here, the blue line represents the removal of oxygen at a very fast rate. We did that for photons. And here on the left, on the right are two slightly different levels of experimenting. The x-axis represents time, and here is the x-axis represents dose. How much dose did I need to give to remove all the oxygen that was present in the medium? And we did that for carbon ions. And in the case of the carbon ions, we even saw the spill structure. So you can see the little steps here represents the different spill structure. That's the different spill structure coming out of the synchrotron that we could even see. And of course, if we do that for time or for dose, we see a different behavior, of course, that is important. So what did we see? We saw that, in fact, oxygen is not depleted at flash rates. So here represents the change of oxygen as a function of dose for each dose rate. And as you increase the dose rate, which means you go on the x-axis, you remove less and less oxygen. So this curve basically says the higher the rate, the less oxygen is removed. And we saw this curve for all three types of radiation, for photons, for protons, and for carbon ions. So literally, we basically show that the oxygen depletion hypothesis, which is the main driver of idea for flash, is not possible. It's not possible to deplete oxygen during flash. I will not get into the chemistry of it. I will just jump. And so ultimately, we even tried to see how much oxygen is left. And we found that at extremely high rates of dose, that we deplete less oxygen than at standard rates. So here is the black line, which represents protons. If I deliver at 1 gray per second, which is close to standard dose rates, I deplete 0.5% of oxygen. But if I deliver the protons at a much higher rate, I deplete less oxygen. So oxygen is not depleted during flash, was the immediate conclusion from this work. And so we basically were able to prove that oxygen depletion does not occur. In fact, oxygen that is there during flash, and it doesn't go away. And so no oxygen depletion for a delivery of 10 gray, and in fact is more oxygen present than the normal rates. Okay. Well, why does flash protect organs and not affect tumor control? Well, no one really knows why, but flash does protect organs. And a question that's still hanging, I'm putting out these questions so that you can think about them. All clinical applications have been using only one beam. Okay, electrons of protons. The second question, which I think you can think about, is the problem of multi-beams. No one has ever really tried to figure out how do you do multi-beam flash. It's an extremely hard question to deal with. Let's see my time. Okay, so now I want to enter another domain that has become a very hot topic in radiation therapy. It's called mini beams or microbeams, and it also allows you to protect organs from radiation effects. So what is mini beams? So in a conventional radiotherapy department, when we treat a uniform region like this. So you see a uniform square, and you see the dose distribution is uniform during treatment. In mini beams, you don't deliver a uniform dose, you deliver a fractionated dose, which is spatially fractionated. So you have a peak and a valley, and it's like a seesaw, right? And so you have these so-called peaks and valleys regions, which you deliver through the whole cancer. So you have a high area and a low area. And the so-called peak to valley dose ratio is the intensity of the peak to the valley. And usually you also have to know the so-called center to center distance, which is usually defined as CTC. So the amazing thing is that if you deliver radiation in the so-called mini beams or micro beams, you can deliver extremely high doses with very little side effects. You can deliver a few hundred gray in these peaks with very little side effects of these valleys to the organ. And this is almost unheard of. Here's an example of a study that was done more than 60 years ago in 1961, where they delivered 140 gray to a millimeter of space, and they literally obliterated this tissue. And by just reducing the beam size to a tiny 25 micrometer, they were able to give 4,000 gray without too much side effects. Look at the difference in the side effects. Here, there is nothing left. The tissue has been literally obliterated with 140 gray. While here at 4,000 gray, which is a factor of 20 or more, you see still a lot of cells in this region. So you see cells here. So the tissue is there and it's not obliterated. And so this was an amazing thing that kind of set off the field of microbeams. And in fact, they found that there was so-called an inflection point also beyond which the tissue became super resistant to radiation. And this is the field size. So here is 1 millimeter, the one on the left, 0.3 millimeters, 0.05 millimeters, so 50 micrometers, and this is the 25 micrometers. And they saw that there was a point around 50 micrometers where the cell suddenly became extremely resistant to very narrow beams, to the so-called microbeams. And so this set off the field, and a lot of people have studied this in the last years. And here's an example of the radiation of a brain, a piglet cerebellum. And you see the stripes, and these are 25 micrometer thick stripes with a distance of 200 between each one. And the brain is totally functional, and they gave close to 600 gray. So you give 600 gray to any cancer, the cancer will be dead. And the person would also very likely be dead. But in this case, there is no damage or very little damage to the mouse, to the piglet brain. And you see the stripes here. So this has been an amazing study that was done many years ago in the early 2000, if I'm not mistaken. And here is a picture of so-called DNA damage analysis, so gamma H2AX. And here is your microbeam, and here's your valley. So your peak is the pink and the valley. And what they see is that everything that is pink represents DNA damage, and you see only the DNA damage is occurring in the peak region, not in the valley. Very little DNA damage is occurring here in the valley. So the difference of giving a homogeneous dose distribution is you produce damage everywhere. But if you use a microbeam, you produce damage only along the microbeam, along the peak of the microbeam, sorry. So possible clinical implementations for this could be you shoot a variety of mini beams or microbeams that eventually blow up into the tumor, and you give a uniform dose to the tumor. And so you spare the healthy tissue here, of course, or you shoot through. In this case, is beams that are shot all the way through the brain, and you treat the cancer that can be anywhere in the middle here. Okay, so some of the most important studies, one of them was done by the group in Paris, Yolanda Pesado, and they studied mini beams where the beams are a little bigger now. They're in the millimeter range, not in the micrometer. And what they did is they studied the shooting of mice, and they took the approach of shooting all the way through, right? So you see the peak to valley ratio is roughly 10. So I have 100% here, and I have roughly 10% here. And I can give extremely high doses in this peak, while very low doses in the valley. And they had a variety of rats that they irradiated, the control, the ones bearing the tumor, the normal rats, and of course, the normal rats with control. And so when they compare the treated versus the untreated, so you see that the untreated is the control here. They die of the cancer, so this is a very rough cancer. While if they treat it, eventually some of them survive the cancer. And the amazing thing is not only are they surviving the cancer, they are cured. So this is a brain glioma for rats that all of them should die, and 20% of the rats are cured. And so this is the amazing results that you have a completely different approach to treating with radiation, where you are literally curing the cancer for a cancer that's uncurable. So it's a bit of a contradiction, but it's amazing that you can cure. So again, we did some work on this to investigate what possible mechanism. I will probably jump on that. But we said that the big difference is that while we tend to look at the physical dose, which is the blue one, what we should be looking as the chemical effect of that dose. And that chemical effect is some chemical. In our case, we proposed that it was hydrogen peroxide, that the chemical is what's filling this gap, and that's what's having an effect on all the cells that are in the middle. Okay, so you see here, I'm giving very low dose, but the cancer is being cured. So something has an effect on the cancer. So we think it was H2O2 that is effectively changing the cancer. And we studied this for a variety of studies that have been around in the literature. We found 4 studies, the Prezado from 2018, the Dombrowski from 2020, and then there was 2 other studies in the early 2008 done in Grenoble. And what they did is they irradiated different types of mice or rats for different times. And they found that there was a difference in survival, of course. And the amazing thing is one of them, this so-called Ragnar study, found very poor survival in one set of animals, while he obtained a 50% survival in another one. And they could not understand why that was the case. So here, the red one was the ones that have the largest distance between the peaks, the so-called 200 CTC micrometers, and they had basically no survival. All the mice died from the cancer. But if they decreased the distance between the 2 peaks to a factor of 100, the survival increased to 50%. So it was a huge increase. In our case, we managed to explain that and show that it's potentially related to this so-called chemical effect. The H2O2 is creating a link between the 2 peaks, and it's ultimately saving the mice from the cancer. So here is the mice that was saved. 50% of them had survived the cancer and in fact ultimately were cured, while all of these, none of them survived and they ultimately died out. So microbeam, presently, no one really understands why they are used, and at the moment the technology for it to go into the clinic is still a bit far away, but it can have a major impact in some of the hardest cancers, like in the brain, when you have gliomas and other types. And it's also amazing for sparing organs to radiation side effects. And with the new pencil beam scanning systems, it will be really easy to develop mini beam approach. So you can just put 2 pencil beams next to each other with a small distance, and you can develop a very simple microbeam or mini beam approach to treating cancer. So it's a potential that is sitting there, that once we understand the mechanism, that we can use for pencil beam scanning. So it's something that could be really advantageous for radiotherapy to look at. So I'll leave these open questions for later. You can follow them later. And so I think that's the end of the talk. If you have any questions, please do. I know it's immediately after lunch, so I hope people are still awake, because after eating lunch, I usually am very tired.
It was a very interesting presentation, Joao. Thank you very much. So we are going to post we have to share the document with the questions. Yes, very good.
Okay.
Can you see my screen?
Yes, very well, Damir, go ahead.
How do those who live in good environment lead a healthy life have to? So I'm not an epidemiologist, so I'm not the best person to ask, but from what I understand, there are 3 main reasons for cancer. One of them is genetic, okay? Another one is environment, of course, and the third one is your personal habits, drinking, eating, smoking. So I don't know which one is the most important, but I know they all have a major role. So it's not only a genetic effect, cancer. For example, lung cancer is highly dependent on the environment where you work, if your lung is under stress, if you smoke, and these are a variety of other cancers could be related to genes and others to your habits. So you really have to pay attention to your own habits. That's also something that's important. Is it possible to achieve flash dose rate at break peak with a cyclone-based facility for in all? So a cyclotron-based facility is absolutely perfect for flash for protons. A synchrotron facility is not. So we could not achieve at the moment that I know of flash with a synchrotron in Heidelberg or any other synchrotron facility that has protons, like in MD Anderson or other centers. So it's perfectly set for cyclotrons. So flash can be done with cyclotrons. Is it possible to achieve mini beams with the cyclotron facility? Yes. The key point of the mini beams is to separate your pencils enough to have that so-called mini beam distance. So the CTC distance has to be at least 1 millimeter. So you can do it with any type of beam. You don't need a cyclotron. You can do it with synchrotron or cyclotron. You just want to be able to separate your beams. Oh, a new question on number 3. No, so LINAC, so I answer a question that was asked in the morning. Is it possible to do flash with a LINAC? The answer is no. Are there photon flash beams out there? Yes, but not in a LINAC or hospital facility. They are only available at the high synchrotron facilities like the Grenoble. Okay, they are not available in any photon therapy facility. So that's why you do flash with electrons and protons, but not with X-rays. And the reason is the target. That target is not prepared for flash. It would literally melt with the amount of intensity. Okay, IMPT, do these examples show implement arc therapy? No, I did not discuss arc therapy. So the concepts of arc therapy are primarily the same as IMPT or IMRT. The only difference is in you spread out the dose over many, many more angles. That's the huge advantage. Instead of using 5, 6, 7 angles, you have more angles. So it's nothing much more. So the main difference for SFUD to IMPT is the uniform, yes. So SFUD are always deliver a uniform dose per beam. The reason that is better than IMPT is that it's robust to uncertainties. So one big problem of protons is the uncertainties, and that's the very reason sometimes we don't use IMPT. But SFUD is robust to uncertainties, and so that's the advantage. What is the time structure of an ion or proton beam? So at the moment, the time structure is something that is under discussion. Ideally, you want to deliver the dose, let's say a minimum of 10 gray in a dose rate of 40 to 50 gray per second, or preferably higher if possible. Okay, so I'm on question 9. What is there a way of increasing the oxygenation cells of some of these hypoxic cancers? No, there is not so easy way to do it. I think most of you have heard of Lance Armstrong, the famous cyclist. He was famous because he won the Tour de France 7 times. And eventually they proved they used a so-called drug called EPO, right? EPO. And this is a drug that literally increases the amount of hemoglobin you have in the blood. And the more hemoglobin you have in the blood, the more oxygen you have. That way you could increase the oxygen content you give to yourself. However, by increasing the oxygen content, you make the cancer grow faster. So you see, it's not a good thing to increase oxygen. So you have to really find a way of affecting the cancer without increasing the cancer kill with the oxygen, without affecting the cancer. So it's not easy to increase oxygenation of cells. You can do it chemically, and that would affect both the cancer and the healthy tissue. If carbon has more fragmentation and destroys the DNA more as compared to protons, then why are we working with carbon ion? Okay, so why are we not working? So the main reason is cost. A carbon facility costs about 5 times a proton facility. And the second reason is there are no clinical data showing a major advantage of it. So there's very few problems, very few clinical studies that have shown the huge advantage of carbon ion. So it's still, let's say, ongoing clinical studies to show the advantage. When I talked about dose rate for flash, I was talking about average dose rate, not instantaneous dose rate. Yes, the instantaneous dose rate is the dose given in a single pulse. I was not talking about that. I was talking about average dose rate. Why do researchers focus on using electrons for flash? Well, every LINAC in the world can do flash because of electrons. They are literally prepared to do flash. That's why every center is using flash for electrons. It's relatively easy to do. You just have to customize the dosimetry. So the big problem of an electron beam for flash is the dosimetry, but you can immediately just press the button and it runs. A LUNAC cannot do photon flash at the moment, okay, because of the Bremsstrahlung target. It does not work for flash because it gets melted at very high dose rates. As current treatments are fractionated, could this have the effect of making cells for certain cancers more radio resistant or tending towards a more hypoxic? So the fractionation is one way of oxygenating cells effect, not the other way around. So the fractionation helps us oxygenate because we kill the cells which have most oxygen, and these, when they die, allow for the oxygen to diffuse through to reach the inner part of the volume. Okay, so if you look at this as a donut, you kill the outer part of the donut, and the inner part then gets oxygenated, and you go step by step like this, and this allows you to, by fractionation, you can oxygenate the tumor. And so usually it helps for oxygenation. So no, number 14, the question is there evidence about dysfunctional mitochondria in cancer cells? In fact, there is evidence of the opposite. The Warburg effect, initially Warburg himself in 1957 gave a famous lecture where he said that the mitochondria is dysfunctional. That's why the Warburg effect happens. He was wrong. It has been proven now in many, many cancer types that the mitochondria is completely functional. There is no problem with it. It just stops using the oxygen for some reason. So there is some switch in the cell that says that the mitochondria doesn't need oxygen. So, but it is completely functional. There's no problem with it. So what particles do you use for mini beams? You can use any type of particles. You can use protons, like the studies from Yolanda. You can use X-rays, like the study from Grenoble. You can use very high electron energy, VHEE electrons with very high energy. You can use carbon. You can use anything for mini beams. You just have to separate them between a few millimeters. You have to separate the peaks. How is the mini beams produced? Well, there are different types of ways to produce it. One way is to use very thin collimators. So you can have a tungsten collimator or a tin collimator which separates the two beams, or you can use a pencil beam scanning approach where you switch on and switch off every third. Okay, so there are different ways to produce mini beams. The most standard approach is to use a collimator which you have a very thin layer of open. So are there advantages of combining protons in different species? There are some advantages, and that's what people are looking at. The main advantage is to do with the RBE. Protons have a low RBE and are very sensitive to oxygen. Carbon has a very high RBE and is not sensitive to oxygen. So what you can do is a form of dose painting. You treat the hypoxic regions with carbon, and you treat the non-hypoxic regions with protons. The problem of ion mixing is time. If you're going to mix beams, you have a problem of time. You will take a lot longer in the treatment room to do this whole treatment because it's a highly complex set of things, right? And you need to have both ions present in the same machine, which is also not common. So it's a matter usually of time, not anything else. So the mixing of ions is usually to take advantage of the RBE and the oxygen potential of carbon, but it's difficult because of timing. It takes too long. So question 18, you mentioned that cyclotrons are perfect to get flash dose rate for low energy. I don't think that it's possible to achieve flash dose rate at low energy beams. What do you mean by low energy beams? You mean like 90 MeV per nucleon? Is that what you mean? Because the flash rate just depends on how much protons I give per unit time. It doesn't really depend on energy. The energy is only important to get to the tumor. So I want an energy that is sufficient to reach the tumor, and at the same time, if I reach the tumor, I can flash to protect the organs that are in the entrance channel. So the energy is only important to see if I get deep enough to reach the tumor. That's why I use that. So it's not really an energy problem. It's a dose rate problem. Okay. Did I miss something, Damir? Because you were adding and then switching up and down.
No, I think that's all. Okay, but we have one more question.
Okay. What is the difference in spontaneous cell death? Okay. So I think it's important that you remember the difference between necrosis and apoptosis. Okay. So let me just explain what they are both. When you take a little knife and you cut yourself, right? Your body reacts to this and it has an inflammatory response. So it sends a bunch of cytokines to the point where you cut yourself to protect you or to react to the situation. So this is an inflammatory response of the body to the effect. And when this inflammatory response happens, it's like basically sending the army to a point to fix the problem. All right. This is really bad if you were to do radiation. An inflammatory response to radiation would kill the person. If you're not aware of how dangerous that is, I would suggest that you read a little bit about the COVID virus on all those lung problems that people have had. Those lung problems are related to inflammatory response to the virus, right? When the body reacts to a stress, it reacts through the so-called army, which is the cytokines, and this is called an inflammatory response. So, and this inflammatory response occurs when you have a necrosis cell death, when the cell literally explodes, okay? In normal radiation therapy, we don't want the cells to explode. We want them to slowly suicide. What is called a programmed cell death is a slow suicide. They slowly get digested and nothing happens. And in this case, it's called apoptosis or mitosis. There are two types. But eventually the cell will slowly digest and disappear. It doesn't explode and throw its contents into the neighbors. And this is really important because the radiation that we give is so little that we don't trigger inflammatory response, but we trigger cell death, right? So the cell dies just by saying, okay, I cannot live any longer, I wish to die. And it sends a signal saying I'm gonna die. And this is what is called programmed cell death. It literally suicides itself. And it doesn't produce any inflammatory response. But it has to tell the body that it's going to die, or else the body will send its army to fix the problem, which is not a good thing. What is could Lear machine at CERN be useful test facility? I would leave that question for people at CERN. I am not at CERN. So I'll leave that question for someone else.
Okay, that's it, I guess.
Yep. Well, thank you
Okay.
once again.
One moment more. There is a question from the auditorium in Egypt.
Okay.
Mahmoud, you can ask the question. You can unmute. Hello.
Oh, okay, Uta.
We can hear you. Yes, please go ahead.
Okay, Mariam she is a student of mine. Has one question, please. Okay.
Yeah, please go ahead.
Please, Mariam.
Hello.
Hello.
And thank you. As I catch from your lecture, I have a question. Does the flash radiotherapy depends only on exposing the tumor to an enormous amount of radiation, of any radiation type?
Mahmoud, could I ask you please to repeat the question because it was very faint. We could hardly hear.
Okay, Uta.
Yeah.
Does the flash radiotherapy depends only on exposing the tumor to an enormous amount of any radiation type?
So yes. So I see a background. So the key idea of flash is that it's not about the tumor, but it's about. Can I ask you to switch off because there's some feedback, please? Okay. The key idea about flash is not about the tumor, but about the organ at risk. So the tumor is not affected by flash, but the organ at risk is protected by flash. And the reason is you give all this dose in one instant. So imagine I'm giving 10 gray in a fraction of a second, let's say a microsecond. For some reason, this large amount of radiation seems to spare the organ to radiation damage. And no one really understands why, but it's amazing that it happens, right? So if I give the same 10 gray over a few minutes, I destroy the organ. But if I give it in 1 millisecond, the organ is protected from radiation damage. The reason is no one knows. That's the big question at the moment in the community of FLASH. And it's really important because it means I can basically get rid of radiation side effects if I do that.
Okay, thank you. Are there any other questions from the auditorium?
Okay, thank you, Yuta.
Okay. In fact, we finished earlier, 15 minutes. Let us continue with the next presentation. I see Hans-Peter and Niklas, they have joined.